No treatment is currently available for traumatic injuries to the brain, spinal cord, and optic nerve in the central nervous system, or the CNS. But our teams at CityU has identified for the first time a small molecule called M1 that can restore the visual function in the mammalian CNS. This molecule offers hope for patients with optic nerve damage, such as glaucoma-related vision loss. To recover visual function after injury, axons of injured neurons must regenerate through the optic nerve. They relay nerve impulses to visual targets in the brain via the optic nerve for image processing and formation. M1 stimulates the fusion and motility of mitochondria. Given that mitochondria is the powerhouse of a cell to generate energy, M1 treatment induces robust axon regeneration by enhancing the intrinsic growth capacity of injured neurons. M1 treatment sustains long-distance axon regeneration from the optic chiasm to the visual targets in the brain. Regenerated axons elicit neural activities in the target brain regions and restore visual functions. We found that the degree to which the injured eyes of M1 treated mice could react to light was similar to that of uninjured eyes after optic nerve injury. We also assessed the response of the mice to a looming stimulus. Looming visual stimulus is a visually induced innate defensive response to avoid predator. Strikingly, half of the M1 treated mice responded to the looming stimuli by rapidly freezing and hiding the shelter, as shown in the lower left panel. We believe that a readily available, non-viral therapy could address unmet medical needs in accelerating functional recovery within a limited therapeutic time window after the CNS injuries. Our results has been published in a high-impact scientific journal, namely Proceedings of the National Academy of Sciences. We are now developing an animal model for treating glaucoma-related vision loss using M1, and that could be used for treating other common eye diseases and vision impairments such as diabetic retinopathy, macular degeneration, and traumatic optic neopathy.